Hello everyone, welcome back to Studious students. Today we are beginning our two-part series on cholera. In part 1, we'll understand the organism, classification, pathogenesis, clinical features, and epidemiology. In part 2, we'll cover lab diagnosis, identification test, and cholera vaccine. So without wasting any time, let's begin. Let's begin with the introduction. Cholera is caused by Vibrio cholerae, which is a curved gram-negative bacillus. It is actively motile because it possesses a single polar flagellum. Its characteristic comma shape makes it easy to recognize under the microscope. The first classification is based on salt. There are non-halophilic vibrios which grow in about 1% salt. Vibrio cholerae belongs to this group. The second group is halophilic vibrios which require around 7 to 10% salt and grow well in high salt environments. Now let's classify Vibrio cholerae based on its somatic O antigen. Although there are more than 200 serotypes, the O1 serogroup is the most important. It's responsible for all pandemics and most epidemics of cholera. This serogroup is agglutinated by O1 antiserum. Another important serogroup is O139, which was discovered in 1992. By that time, 138 O antigen serogroups had already been identified, which is why it was named as O139. This serogroup has mainly been reported from the coastal regions of India and Bangladesh. Now let's look at the non-O1 and non-O139 serogroups. This strain belongs to neither O1 nor O139. According to notes, they have not caused epidemic cholera so far. This makes them less significant in causing large outbreaks. Next is biotyping. There are two biotypes, classical and LTOR. The classical biotype caused the first six cholera pandemics and is considered highly virulent. The LTOR biotype caused the seventh pandemic and was discovered in LTOR, Egypt. Based on serotyping, Vibrio cholerae is divided into Ogawa, Inaba, and Hikojima. During epidemics, Ogawa can shift to Inaba due to changes in the RFBT gene. Hikojima is considered unstable because it expresses features of both Agawa and Inaba. Let's quickly summarize the classification. We start with the Vibrio genus. Within it is Vibrio cholerae, which is divided into O1, O139, non-O1 slash O139. The O1 serogroup is further divided into the classical and Altor biotypes. Finally, these are classified into the serotypes Ogawa, Inaba, and Hikojima. This flowchart makes the entire classification easy to remember. Now let's begin with the pathogenesis. Cholera is transmitted through contaminated food and water. Since Vibrio cholerae is heat labile, a large infective dose is needed. Around 10 raised to 8 bacilli is required to survive the acidic environment of the stomach and to reach the intestine. Once it reaches the intestine, it can establish infection and produce disease. The whole events are happening in the intestine. Cholera toxin binds to GM1 receptor. It then enters the cell by endocytosis. It moves to Golgi apparatus to the ER. A subunit is released into the cytoplasm, as you can see in this image. It then activates the GS protein by ATP ribosylation. Adenylate cyclase remains continuously active. Intracellular CAMP increases. Protein kinase A is activated. CFTR chloride channels open. Chloride ions are secreted into the intestinal lumen. Water follows by osmosis. Profuse rice water diarrhea develops. The incubation period of cholera is usually 1 to 2 days. Patients typically present with profuse watery diarrhea, often described as rice water stools. They may also develop vomiting, which further increases fluid loss. These symptoms can rapidly lead to dehydration if not treated. Because of severe fluid and electrolyte loss, patients may develop muscle cramps. Continuous loss of body fluid also leads to significant weight loss. Without prompt fluid replacement, these complications can become life-threatening. Let's take a quick look at the history of cholera. Since ancient times, the Ganges, Brahmaputra Delta, and Bangladesh have been considered the homeland of cholera. Many major outbreaks are believed to have originated from this region. The first six cholera pandemics, which occurred between 1817 and 1923, were caused by the classical biotype. These pandemics began in Bengal and gradually spread across many parts of the world. The seventh cholera pandemic started in Indonesia. Unlike the earlier pandemics, it was caused by Altor biotype. Altor produces more carriers and is less virulent than the classical biotype. The Bengal strain O139 is considered to be a derivative of Altor O1 but differs in being encapsulated. It may be the next big bad boss, but luckily O1 Altor strain remains dominant, limiting the spread of O139. Now let's discuss the epidemiological determinants. Humans are the only reservoir of infection. The source of infection may be asymptomatic individuals or carriers. Transmission is highest during periods of high temperature, heavy rainfall, and flooding, often referred to as the cholera season. People with blood group O are at a higher risk of developing cholera. Vibrio cholerae is acid labile but remains stable in alkaline conditions. It is also heat labile, although it survives well under refrigeration. Drying and direct sunlight can destroy the organism within a few hours. Thank you for watching. If you found this video helpful, don't forget to like, share, and subscribe to Studio Students. See you in the next video.